I think I will give a different type of talk. What I will talk to you are some unpublished data that we have collected in the past few months in the lab on the modulation of murine endogenous TDP43 by the glutamatergic system. As a member of a ribonucleoprotein family that has an emerging role in synaptic, in regulating synaptic plasticity, TDP43 may well participate in synapses formation and stabilization, and may play an active role in synaptic plasticity. We know, and we have heard in these days, that TDP43 can exit the nucleus, travel along the axon, reach the terminal branches packed in ribonucleoprotein complexes. So the question that we asked in this study was, is TDP43 mRNA also localized at the dendrites? And if so, is modulated by synaptic activity, specifically by the glutamatergic system? What we know already is that neuronal depolarization can modulate TDP43 localization within the neuron, and can enhance its localization to known RNA binding proteins like FMRP and Stauffen-1, these in hippocampal neurons. Is also modulated by other factors like BDNF in primary cultured motor neurons. Our data on cortical neurons are similar to the data in hippocampal neuron, and we also found an increased co-localization of TDP43 with PSD95 under potassium chloride stimulation. So we wondered if another type of neuronal depolarization induced by the glutamatergic system is modulating TDP. This cartoon represents a glutamatergic synapse with the 4 receptors that released glutamate can activate. We have focused our attention on the NMDA receptor because many types of synaptic plasticity rely on this receptor. First, we look at the protein localization. These are in cortical culture, primary cortical culture from mouse. And I don't know if you can see, but is real. We observed a nucleus to cytosol movement of TDP43 under glutamic acid stimulation in the first 5 minutes of the treatment, and an increased co-localization of PSD95 with the synaptic protein of TDP43 with PSD95 in the neuronal dendrites, again after under glutamic acid after 40 minutes. The next question was, TDP43 mRNA is localized at the dendritic spine. Local translation is a very important phenomenon in a polarized cell as the neuron. It allows the tagging of specific synapses and the fast translation, the fast production of protein needed during synaptic activity, so during synaptic plasticity, for synaptic plasticity and spine morphogenesis. we prepared synaptosomes from cortex, from mouse cortices. And here is just to show our preparations with the enrichment of the synaptic proteins, synaptophysin and PSD95. And in this synaptosome preparation, we have amplified TDP43 mRNA and also another ribonucleoprotein A1 and another ribonucleoprotein mRNA A1 known to interact with TDP, and in polyadenylation experiment that we have performed, we have observed a polyadenylation induction of A1, so under potassium chloride depolarization. So we thought that A1 could have been a good candidate along with TDP43. First, we verified the synaptosomal activation under NMDA stimulation. We did that looking at alpha-cam kinase phosphorylation, which is a very established target of synaptosome stimulation, and we observed a significant increase under NMDA. From these stimulated synaptosomes, we purified the polysomal and subpolysomal fractions, collected them, and amplified with real-time PCR. These are two actual absorption profile on a sucrose gradient of a total cortex preparation and synaptosomes preparation. Usually the polysomes preparation are before the 80-60 subunits of the ribosomal components. The synaptosomes have much less material, so it's difficult really to discriminate these components. And we observed calculated as PMP percentage of target mRNA on polysomes, an increased localization of TDP mRNA under NMDA stimulation on the polysomal fractions, meaning that indeed TDP43 mRNA is not only present at the synapse, but is also actively translated during synaptic activity. On the other hand, at least in this condition, A1 did not localized in the polysomal fraction. So in this condition, A1 is not actively translated. And our working hypothesis is that at the synapse, there are probably two, I don't know how to call it, pools of TDP43 protein, one cytosolic arriving from the granules, and the other synaptic, meaning that is translated on site. And the hypothesis, our working hypothesis, that TDP43 being synthesized also at the synapse is well positioned to be an active player in synaptic plasticity. We are currently doing long-term potentiation experiments to check if this is indeed the case. On a broader view, there is the hypothesis of a synaptic degeneration leading to neuronal cellular degeneration is gaining momentum now in the neurodegenerative field. And I just want to acknowledge Michele Nutini, that has done all the experiment that I have talked to you about, and Stefano Loreni and Sara Caldarola at the University of Tor Vergata in Rome, that have helped us in setting up the polysomes preparation and the funding agencies. Thank you.
Thank you, Patrizia.